Hello everybody, now we'll take up the structure of nucleus today. Nucleus, the director of the cell, very important structure with chromatin inside. Chromatin is the DNA network which lies scattered inside the nucleoplasm. Here the nuclear envelope is there, that is a limiting boundary of the cell, and here where the inner membrane, see we have two membranes, the outer nuclear membrane and the inner nuclear membrane, wherein the inner nuclear membrane fuses with the outer nuclear membrane, pores are produced. They are referred to as the nuclear pores. The outer nuclear membrane is the rough membrane, because it has ribosomal content over it. The ribosomes are studded on it. The inner membrane is smooth one, because it has no content of ribosomes. In the center, we have a non-membrane bound structure called the nucleolus, through which all this chromatid network is redeemed, it is created. That is, nucleolus has a lot of rRNA, because of which this rRNA, DNA, because this chromatin network, it is composed of DNA, rRNA. So basically rRNA is produced from nucleolus. So from here we can connect these fibers with the nucleolus. That is, these are actually, they have origined from this nucleolus only. In the cells where the protein efficiency must be there, this is very common. This completes with the structure of nucleus. There is not no importance if we deal with the structure. Basically it was discovered by Robert Brown, which we had discussed in the first section also. Robert Brown. It was Fleming to give the term, Fleming to bestow the term chromatin, because it is stained with basic dyes. Also to mention the structure, see, I'll help you out if you have not understood. See, it's a very simple structure, fine. So that is why I had drawn such a simple structure, because it indeed was a very simple structure. So I have not anonymously made anything arbitrarily chosen, but it is, yes, the structure is that simple only. Wait a second. This is the structure: nuclear pore, chromatin, nucleolus, nucleoplasm, and nuclear envelope. Now you compare these structures with this one. This structure was also the very same: nuclear pore, nucleolus, nuclear envelope, outer nuclear, inner nuclear, chromatin, perinuclear space. Yes, perinuclear space is between the inner and the outer membrane. Fine. So this is the structure. And let me tell one more thing to you. Here very important, anucleated cells, they are at maturity, such as mammalian RBCs and sieve tubes, they do not have any nucleus in their cells. We have many which have multinucleated cells. They are opalina. The binucleated cells, examples paramecium. If we talk of, yes, one more thing I may mention, that chromatin fibers, except from DNA, RNA, or anything like that, it even has certain proteins referred to as histones. We'll talk of the function of histones in the later part. Just you need to know that histones, they also are present in this chromatin part only. Now we have different types of chromosomes. I hope that you are aware of chromosomes. We have acrocentric, submetacentric, metacentric, or SAT chromosomes. See, this way you won't understand. Let me tell you in my way. See, when the chromosomes, this part which bifurcates the two loops, it is referred to as the primary construction or the centromere. This centromere, when it is at the center, it is called the first part, that is the metacentric chromosomes, in which the centromere is at the center. When the centromere is near to the one loop, somewhat near to, not at the center, but somewhat near to one loop, then it is referred to as submetacentric. What do we refer to as acrocentric? The chromosomes in which a particular kinetic centrosome is very near to one loop, then such sort of structures are formed. What about telocentric ones? Telocentric ones are the ones in which it is at the apex only. The centromere is at the apex, so that no second loop is formed. So this type of structures can be redeemed. More to know, you can deal with your NCRT. It talks of what happens in different phases. That we'll deal later part when we study the cell cycle chapter. There will be talk of this. Now SAT, this is no importance if we talk of it now. Basically next part will be microbodies that I'll deal with in the later section. Thanks.